Hi, thanks, Kayla. Welcome, everyone. Welcome to the first GFRA webinar of 2025. I'm tasked with introducing Michiel Harmsen on behalf of GFRA, and it's a real pleasure to introduce Michiel. Michiel's been studying biology since 1988, and it's really great to to introduce this seminar, this webinar. This is a subject that Miguel has pioneered. The use of antibody reagents and especially single chain reagents to understand the FMDV particle and to apply that to vaccine quality. And this is something that there's a real growing interest in over the last decade, and something I'm very interested in recent years. But Miguel was the person who started all this, and really is the person who's the famous person for this in the world of FMD. So really looking forward to your webinar, and over to you, Miguel.
Thanks, Toby, for these kind words. Kyla, could you, for sharing my presentation, I press share, I assume?
Yes.
And then I have to share screen too. I think I should be visible now. Is that true?
I'm not seeing it yet.
And I'm going to say share then.
There we go.
It's visible now?
Yes, it is. Thank you.
Okay, good. Then I have to get, I'm not sitting behind, I was to get my mouse here. Okay, good. And I have to, can everybody see my mouse moving also?
Yes, we can.
Okay, then I will simply use that as a pointer. Okay, thanks. Sorry for the delay. I was having trouble getting in. It was not due to Kyla or Toby, but just due to me. I'm happy I'm in. I'm happy to give this talk about a topic I'm busy with at WBVR with my colleagues for about 20 years. It's focusing on analysis of foot and mouth disease vaccine quality using single domain antibodies. I think I have 1/2 hour, Kyle, and please interrupt me if it's getting too late. I'm afraid I'm got a slightly bit much of slides, trying to squeeze 20 years in. So I'm going to skip a few also, I think. Okay, now I have to figure out how to, that's it. This is where our institute in Lelystad is present in the Netherlands. This is WBVR. It's in Flevoland, which is below sea level. You can see our dike here to prevent this from getting flooded. I'm actually now here in this building together with many of my colleagues. Aldo and Feder are often present in this high containment unit. I will not focus too much in this talk on camelid heavy chain antibodies and the recombinant VHH or antibody domains derived of them that we use for the mouse disease research, since I think it would distract too much. For the purpose of this talk, they are just antibodies. They are just recombinant antibodies. And I'm going to only briefly address some aspects of them. And with my next slide, I'm going to give an overview of the published work. This is a complicated slide, quite complicated. I'm going to use quite some time to discuss it. It's a kind of the talk outline that I'm going to give of research that started some 20 years ago after I was busy working on VHHs to develop foot and mouth disease therapy using VHH as passive immunization. And I'm not going to address the couple of papers we published on that because I think it's not really feasible to use in practice. But we got a nice panel of foot and mouth serotype O binding VHHs, including these three that are present often in my talk, that are nice. And that's where I would like to start. And what we did is we first developed some methods also to coat these VHHs, which are small antibody fragments. They are not complete antibodies. They are antibody fragments. To coat them in a functional way to polystyrene plates, you need this long hinge with a cysteine tag attached to it to do it well. This is the work I did in blue, and in orange are the papers, the overview of the papers published on this topic of vaccine analysis. We then analyzed, some 50 years ago, I started collaborating with our factory that we still had in those days that was not sold yet to Michiel, now Boehringer. And we started collaborating to analyze vaccine stability, and we used these VHHs to do that. We also use cell-deTOF-MS, which is a mass spectrometry method to analyze foot-and-mouth disease antigens. And that was highly instrumental to determine that actually 12S was one VHH was highly specific for 12S. That was really a nice discovery. And then we later discovered M170 being 146S specific, and we used these also then these ELISAs quite nicely to analyze vaccine stability and show that thiomazole destabilizes and BSA and sucrose stabilize foot-and-mouth disease. Since these 146S specific VHHs are highly serotype specific and often also highly strain specific, we then wanted to have more of them against other serotypes, Asia1, SAT2, and serotype A, that is published. And we recently, that's unpublished, also isolated better serotype O binding VHHs that recognize most serotype O strains. And we also, in the last couple of years, especially in collaboration with several Chinese collaborators, did mapping of antigenic sites of these particle-specific VHHs by cryo-EM, typically Chinese work, and also by cross-linking mass spectrometry. Okay, I've just given conclusions now in a high-end overview. First, some examples of the use of VHHs for vaccine quality control. This slide shows, I'm not going to introduce foot-and-mouth disease in detail. I hope the audience from the GFRA knows what foot-and-mouth disease is, and that you can have serotypes, the 7 serotypes are known, and that you also know that briefly that a complete virion looks like this. Sediments at 146S in sucrose gradients. You can make VLPs, typically work that Toby and people from Purbright and MSD can talk a lot more about. You can make virus-like particles that lack the RNA genome, that sediment at 75S. You also have natural empty capsids. And when you do mild heating or mild acid treatment of these 146S or non-stabilized VLPs, then they easily dissociate into 12S pentamers. And this is a big topic of my research that I would like to address in this talk a lot. And the point is that this is a good vaccine. It should be intact. And if this happens, this dissociation into 12S, it's lowly immunogenic and you lose it. So that's why these particle-specific VHHs against 12S and 146S are valuable. So we've got this M3, which is 12S specific and recognizes most serotypes. And we've got these many different panel of VHHs against different serotypes of foot-and-mouth disease. Now, the old method to measure, the classical method to measure amounts of 146S particles is a simple method based on sucrose density gradients that was published 50 years ago by Simon Bartelink and Rob Malone from our institutes. It has one figure, this paper, and that figure is shown on this slide. And what you do is then simply you make a sucrose cushion gradient, you put your sample on top, you centrifuge and ultracentrifuge, and then you measure UV absorbance. And if you do not do RNase treatment, you can see the 146S peak here, and the area under the curve is a measure for amount of 146S. And that's a bit of a laborious method. Because you cannot then analyze a lot of samples in an ultracentrifuge, and also you don't see VLPs that would sediment about here because they don't have RNA. So you cannot identify them by UV absorbance. So now to VHHs. This is one example. I've just mentioned it earlier about the first discovery about this particle specificity of M3 for 12S. This is a cell-Titov experiment where you do an immunocapture of foot-and-mouth disease antigens with either M8 or M3. And then the antigens captured are detected by mass spectrometry. And with M8, it binds both 12S and 146S. You can see the VP123 peaks of the capsid, and also the VP4 protein that's more internal in 146S particles. While with M3, you can still see VP123, but you do not see the VP4 peak. And that was, luckily, we realized that implied that it was specific for 12S because 12S particles do not have VP4 protein present. When the particle breaks open, the VP4 is released and separates from 12S. We later then also discovered that M170 is 146S specific, so it's shown on this slide. M3 ELISA, a double antibody sandwich ELISA, only detects the 12S peak in sucrose gradients of acidified antigen, while M170 only detects the 146S peak of the untreated antigen. Well, these are the rather simple schemes for the setup. We always do a sandwich ELISA, coats with unlabeled antibody, unlabeled VHH, detect the antigen with bioethylated version of the same VHH and a streptavidin conjugate, and the same for 146S using M170. Initially, we also had a nice trick while we only had M170 for serotype O to try to detect 146S of all other serotypes. We used the M3 ELISA using samples that are untreated. Then you only detect 12S, and samples that are heated, and which are then 12S plus 146S, because this then is converted into 12S also and detected by M3. And if you then subtract these two figures, you know the amount of 146S in your sample. And if the amount of 146S is a lot, then this ELISA setup works nicely. Yeah, I think I've said it already when discussing this Bartling and Malone paper, that advantages of VHH-based ELISAs for 146S VOP quantification is sensitive, far more sensitive than SDG, high throughput. They are serotype specific, which is good if you want to analyze in multivalent vaccines. It's not nice because you need VHH for all serotypes, and you can also use them for VOPs. Now, this is an example of an analysis done in vaccines. So when you have assays where you can use adjuvanted vaccines and analyze them in ELISA, and this was Owamanissa, that while preparing a vaccine, we retained the sample without adjuvant, so the aqueous solution, the first emulsion made of the same batch, and the complete vaccine, the double emulsion. And we stored it for a long time, these 3 batches, for almost a year at 4 degrees. And then by combined M3 and M170 ELISA, we could determine the fraction 146S at intervals during storage. And you can see that without adjuvant, it's relatively stable. It decreases only late. But with adjuvant, it goes down fast. And you could see in these samples also that 12S accumulated in these samples. So it was instrumental having both a 12S specific and a 146S specific ELISA to see that 146S dissociation occurs in vaccines due to presence of oil. And then later, we've shown that presence of thiamazole, again, example Owamanissa ELISA, is detrimental for integrity of 146S particles. It goes down faster as compared to example without thiamazole. And also we've shown, I've simplified it from these papers, just shown the essence. We've shown that the presence of 30% sucrose and 1% BSA, the green line, then the vaccine is stable. And if you don't add it, it really goes fast. It goes down fast. It's the synergistic presence of these two that's you need them both to have a good stability of this strain. Okay, I'm gonna speed up a bit on this topic to be sure I have time for the last part. Isolation of 146S specific VHHs. This is a kind of acknowledgement also, which I updated slightly, but not perfectly for this occasion. I use this slide a lot internally to show what I would like to call my group, not Aldo and Feder's group, about especially the VHH part. And we have a pipeline to isolate VHHs where we write an ethical protocol, and Aldo and Feder are here helping me in this part as veterinarians. We do Lyme immunization, phage library construction, 2 rounds of phage display selection, screen individual VHH, sequence them, do yeast expression, and then do whatever kind of assay you want to do with an antibody that you're interested in. That I've now focused a bit on foot and mouth disease, VNTs, cryo-EM by Hao Zhou Li in our Chinese collaboration. These are all technicians: Connie, Marga, Sandra, Quillen, Renee, Cynthia, Helmi, that did a lot last couple of years. And at the left are the people that are more doing paperwork. Nishi now is part of our group also doing lots of lab work as a postdoc. I didn't do a lot on foot and mouth disease, but most on other topics. Naive library, although she did some on serotype O VHH recently. Yeah, of course, I collaborated a lot with the foot and mouth disease group, and that should be stressed also. I've only shown Cynthia, but that's of course also an important aspect of this talk. Phage display selection of 146S specific VHHs. So you immunize a llama. We've got them immunized with different strains, different llamas. We take blood samples after immunization for phage library construction, and you do this trick shown in this scheme here. I'm not going to discuss it. It's phage display selection. It's a trick where you isolate your antibody, but also the sequence of your antibody, the DNA sequence, so you can recombinantly produce it and use it as a monoclonal antibody. We did a trick, and I'm also going to be brief on this one, that it's essential that during these phage display selections, these 146S specific VHHs, if you want to isolate them, they are rare. They are not abundantly present in libraries. And if you want to have a good one, which is highly specific for many strains, then it's really 1 in 1,000 that you want to find. So during panning, we also had these tricks to compete with 12S particles to really direct them towards 146S specificity. This slide is a slide showing the reaction of a panel of VHHs with a panel of foot and mouth disease strains that are color-coded according to their serotype. And both untreated antigen in blue and heated antigen, so 12S in red, were measured. And as you can see, M8 binds almost all strains and both 146S and 12S, while these ones here in the middle are all quite 146S specific. You only see blue bars, and they are serotype specific. So these ones bind serotype A. This one is really highly strain specific within serotype A. This one binds Asia 1 and M170 for serotype O, but only 2 strains. This is a slide showing the reaction of VHHs in sucrose density gradients. It's from the serotype A paper of Hao Zhou Li. So sucrose density gradient fractions were made of A22 Iraq and A24 Cruzeiro, and then analyzed all fractions by ELISA using different VHHs. You can see M691F is highly specific for the 146S peak, does not detect 75S here or 12S here, while other VHHs of the purple CDR3 group do bind these antigens. And for A24 Cruzeiro, it's fun to see that you also see a A24 Cruzeiro contains 75S. This peak is 75S. It's an empty capsid. It's a peculiarity of this strain. And you can see that M702 also binds this 75S, which is probably similar to VLPs, and 146S, but not 12S. While M691F is highly specific for 146S. It's an intriguing thing I would like to stress because I have no explanation for this difference. Serotype O, we've got a novel panel of 146S specific VHHs for serotype O. Same setup of slide as shown earlier. M170 was not binding to OSKR and OTAI1, and we've got these two novel ones, M907 and 916, that do bind all these 5 serotype O strains in a 146S specific manner. That's also shown in this slide, similar to the earlier one, sucrose density gradient fractionation. We do have 12S present in these samples in control ELISAs, as you can see here, for these 3 different serotype O strains. And for all 3 strains, 916 and 907, purple and red, you can see only the 146S peak is detected. So they are highly specific. And here is another ELISA showing this, that these 2 novel ones are good. Red line, 12S, not detected at all. Even in a sample spiked with contaminating 12S, you can detect blue line, the 146 as well, equally as the green line that's not spiked. Yeah, I think due to time, I'm going to be short on this one. I'm just going to say we have isolated VHHs with good affinity. We use octets to measure it. This is an example to show that we can do a good experiment with good curve fitting. Affinities are often low. Only with M916, the novel one, we had a bit high KD, 264 nanomolar. That's not a good affinity. KD is too high, and that could be due also to reduction of the disulfide bond. We normally produce VHHs in this format. That's a mixture of dimers due to disulfide bond formation and not. And we think this is essential also, this format, for having good binding in ELISA. And I want to have time, and that's why I've been short in the previous one for this last part, foot and mouth disease structure, antigenic sites, and immunogenicity. This is an introductory slide taken from the internet, from this publication, just to show what it looks. I've shown it earlier. This is a 146S particle, which has these 60 copies of VP1, 2, and 3, the outer part, that are colored always blue, VP1, green, VP2, red, VP3. Try to remember it for this last part of the talk. And these 12S particles are repeated 12 times here in a virion. And there are symmetry axes. And you can immediately see that this is the 5-fold symmetry axis. I'm going to use that term. Try to remember it. So VP1 is close to the 5-fold symmetry axis. And this is the 3-fold symmetry axis. At each of these edges, these 5 edges, there's a 3-fold symmetry axis. And this axis is the 2-fold symmetry axis. This is, you can see here, mirrored VP3, VP2, the same, VP3, VP2, are attached here. This is the 2-fold symmetry axis, is where the 12S falls apart. That's where it's dissociated, when it dissociates into 12S. The definition of antigenic sites that I use is based on the old, very, very old nomenclature used by isolation of monoclonal escape mutants. That's published a lot in 20, 30 years ago. And this is shown here, modeled. 5 sites were initially identified, and I would like to focus only on the initially identified sites and not the refinements of novel VP2 and novel VP3. And I would like especially to focus on sites 1 to 4. And these are indicated here. Site 3 is in ball and stick the side chains of residues that are mutated in monoclonal escape mutants that define this site, which are on VP1, more close to the 5-fold axis. And it's enlarged here in this rectangle. And site 4, the orange balls, are on VP3, close to the 3-fold axis. We're going to see this site more often. It's really the red, VP3, where it's located. Site 2 on VP2. And site 1 is mostly on the GH loop of VP1, which is often flexible and extruding from the surface. Okay, so we collaborated with these Chinese researchers. This started in 2019. This is PhD student Pan Liu and supervisor Xiangxi Wang from the Institute of Biophysics in Beijing, and PhD student Haozhou Li, and I think I should call it postdoc or junior researcher Dong Hu did a lot of lab work, and Hu Chengguo and Xi Sun are supervisors from the Veterinary Research Institute in Lanzhou. They did a lot. They did cryo-EM work in Beijing, in the lab in Beijing. And we're going to show first work primarily done by Dong Hu in resolution of the structure of M170 complex to serotype O and M8 complex to serotype O. And you can see M8 is binding on top of this region, and this is the GH loop. And so I call it site 1, old nomenclature site 1. This is not 146S or not 12 as a specific site. While M170 binds here close to the 3-fold axis, it binds on site 4 of VP3. We also recently mapped these novel serotype O146S specific VHHs by competition ELISAs. This is unpublished. We had these novel, especially 907, 916 clones. And if you do competition ELISA of bioethylated VHHs with unlabeled VHHs, then you can get these blocks of clones that show reciprocal competition. It's not completely reciprocal here, but I think nevertheless it's one group, one site that all these serotype, this 146S specific VHHs recognize. And so, since M170 is site 4, I would say also these novel ones recognize the same sites on VP3 close to the 3-fold axis. Now we also mapped to serotype A, 146S specific VHHs, M702 and M691. We used cross-linking mass spectrometry for that, since we did not want to disclose the sequences of these VHHs. Since we would like to keep them for sale. And this is a technique where you complex the VHH with foot and mouth disease, cross-link it with a bifunctional chemical linker, then do proteolytic digestion and identify cross-linked peptides and actually the residues that are cross-linked by double MS. And here in balls are the side chains that are cross-linked to M702, and here the same for M691. And this structure is superimposed with the M170 cryo-EM structure to show that also for these two, it's the same sites on VP3 the site 4, roughly, this is not an accurate mapping, but roughly the same position where this 146S specific VHH is bound. I'm going to skip this one. And this is a map showing 146S specific VHHs for serotype A, that actually the 146S specific ones bind 2 independent sites, 2 blocks here. We mapped 2 of them, of these different blocks. One of them mapped on site 3, so close to the 5-fold axis, and the other one, this is not a pentamer. Note that this is not a pentamer. This is not a 5-fold, but a 3-fold axis. The other one is very close to the 3-fold axis, so which is again site 4. Haojiu Li and the group from Beijing also mapped the structure of 12S, resolved the 3D structure of 12S particles. And one thing that was found is that it had a different structure. That's here is a superimposition of a pentamer as it is present in 146S in gray, and an isolated 12S, and the structure of an isolated 12S. And what happens is that it's more curved. It's schematically shown here, and the reality is more here. You can see the blue more here, indicating it's more on top, and on the edges you can see more the 146S. It's a different structure. What happens if 12S dissociates, the structure changes. There's a lot more details in the paper about all these changes and where they occur. They also resolved the structure of M3 bound to a 12S pentamer, and noted that it did not bind to the outside surface, but to the inner surface. Here, M3 in orange is binding to the inner surface. It's again VP3, but it's not on the outside, it's on the inside. And that, of course, immediately explains its high 12S specificity. Now, the reasons for being 146S specific are explained in this slide. It was initially suggested already by Hu Dong that based on a low resolution 12S structure that was compared, that was superimposed with the 146S structure, that the epitope where M170 bounds, that's here the balls again, that's changed, that the positions of atoms are not at the same positions in 12S as compared to 146S, and so that this altered 3D structure explains the 146S specificity of M170. And also for M688, this was shown recently that this occurs on site 3, this is the other side, that the positions of atoms is changed in 12S. This is more reliable, this is a reliable 12S structure, is changed, and that this can explain the specificity for 146S. For M678F, an alternative explanation may apply, because here the epitope is split over the interface where 2 pentamers associate. And because the epitope is split, if it dissociates, the epitope is split into 2 halves, and that can affect binding, of course, also. And the same applies to a bovine map, R55. Now, for the last part, hope you're still alive and with me. I think the dessert is the nicest. We did this is an old study showing that, this is unknown for 20, 30 years already. If you immunize, this was pigs, with intact 146S, you get a good neutralizing antibody response. But if you immunize pigs with 12S, neutralizing antibody response is much lower. But if you immunize with 12S and do polyclonal ELISA against a mixture of 146 and 12S, you can see good immune response. So 12S is immunogenic, but 12S antibodies do not protect against 146S. And it was nicely shown in this last paper using VHHs or bovine map or VHHs again against 5 sites, the internal site of M3 and sites 1, 2, 3, and 4, old nomenclature, of these antibodies, is that if you compete these polyclonal sera from these pigs immunized with 146S for class 1, this site, which was a 146S specific site, you can only see competition with after vaccination with 146S, but not with 12S. Class 2, you can see a bit response, not perfect. This is M8, site 1, which is not 146S specific, not very. Here again, a highly specific site and a highly specific site, you don't see a response. So it appears that the explanation for the reduced immunogenicity of 12S particles, of neutralizing antibody response against 146S by 12S immunization is due to this different conformation of 12S particles. Okay, this is my last slide to wrap up. This is I'm going to stop work at WBVR as of 1st May. We will still be able to do llama immunizations and use production of these VHHs by these people. I thank all my collaborators. Vimian van der Wal will take over VHH production in yeast, and Tinker Jelsma, after 1st of May, will take over llama immunizations. I would like to give the word back to, I think, Toby. Sorry not to have discussed the beautiful VP4 work, Toby. Thank you for your attention.
Thanks, Miguel. That was a great tour de force of your work on VHHs for 146S specificity. Fantastic stuff. And yeah, I mean, it's great to see all that in one presentation, and the explanation, the functional mechanism for how those antibodies have their recognition. Really great. So the floor is open for questions. If anyone has questions, please get typing. Do you think there's any chance of the 146S specific reagents being found that are more broadly reactive across a whole serotype, or some of the ones you've got already fill that category? Do you think?
Yeah, we've got one slightly serotype A and C. In the panel described for serotype A, we got one or two showing some cross. First of all, 146S specificity is not. I've shown here the best ones we have that are highly 146S specific, focusing on those because they are useful for many assays. But I think there are also many intermediate, slightly 146S specific antibodies. So your question also has to do with what do you call 146S specific? If you would ask it to be not 100-fold specificity, 100-fold lower, which is high, then I would say that's going to be difficult. If you would say M8 does show some 146S specificity for Asia1 Shamir, 16-fold. Type C, I don't know the figure, but also somewhat, but does bind quite some serotypes. It's fluidic. The answer is not exact to give here. Yeah.
Okay. Oh, hang on. I'm looking in the wrong section for the questions. Here we are. So there's a couple of questions coming in. So this is a question from Laura Constance. Have you used HPLC to compare with the ELISA for characterization instead of sucrose gradient analysis?
And the answer is simply no. We don't have this technology going on.
Okay, that's a simple answer. Okay,
Yeah.
there's another question. An anonymous attendee has asked, thanks, very nice talk. The altered 3D structure of the 12S is for both heat-treated 146S and naturally produced defected 12S particles. That's a question. Does that
If
make sense?
I remember well, the 12S was prepared by acidification. I think our Chinese collaborators chose this way because that's the natural way how 12S is generated in during the infection process in the body by entering the endosome. And so it was a not we normally use heating in our analysis because it's a more simple method.
Yeah.
But the cryo-EM structure was from acidified 12S.
But I mean, presumably the 12S from heating or from acid would probably be the same, you think?
I would assume
Yeah.
that. Yeah.
Yeah.
Yeah. That's I have no reason to believe there's any difference. I have
Yeah.
never seen in any experiment any difference.
Sure. Okay, next question from a name you might recognize, Jose Coco Martin. Thank you for the great presentation, Miguel. During your introduction, you mentioned the 5S particles. Do you have any indications that a 146S particle can dissociate into 12S and subsequent 5S?
Thanks, Jose. Was nice collaborating you with the initial phase. You also played a role in really in the early days of particle-specific VHHs. We never studied it. Again, I have to give a blank answer and say we never getting 5S would require other kind of sucrose density gradients that I think to separate them from 12S, we never analyzed it. I have no clue. Sorry.
Okay, what does M mean?
M is Dutch for mond and clausea, which is foot-and-mouth disease in Dutch. So we started clone numbering based on antigen. M is foot-and-mouth disease.
Okay, that's
Sorry.
nice to know, actually. Yeah.
Yeah, sorry, didn't say that.
Okay, question from Benedicto Biamukama. Why does 146S VHH specific degrade faster in oil, and what are the implications of this for vaccine stability and efficacy?
I'm not sure if I understand the question correctly. I would be. You could help me, Toby. I would be tempted.
I think they're asking why does the antigen degrade faster in oil?
Yeah, of course. That's a logical question. That's a good. Yeah. We also asked. This was a pivotal thing. Jose and I, and when we collaborated, saw in that phase, and we asked ourselves the same and looked a lot in literature, and actually appears that, and that's also in the publication I gave about this BSA and sucrose stabilization, where we describe this phenomenon. And there are a couple of publications that water and oil emulsions are actually denaturing environments. The interface of an oil to water is a denaturing environment, and the inside of a protein is hydrophobic and tends to get unfolded there. And I think that's what's happening. That's why oil emulsions, and perhaps not other adjuvants like aluminum, will not have the effect, but oil emulsions are protein denaturing, and therefore also results in, for some reason, in dissociation into 12S.
I mean, that's an interesting. I mean, I totally agree with everything you said, but why do so many manufacturers use oil emulsions? If the simple answer would be to go back to aluminium aqueous vaccines, is it that simple that they don't degrade so fast?
Of course, nowadays, I cannot say and often I think formulations of vaccines are things that vaccine manufacturers tend to keep as proprietary knowledge. But we did show, and I think also others in literature did show that if you add sucrose and BSA, that you can have really big stabilizing effects. But so these excipients are really important to solve this problem.
Yeah. Okay.
Yeah.
Okay. Next anonymous question. How do you think about your 146S specific VHHs? Are they equally also recognizing 75S VLP derived from the same serotypes?
Look at figure 6 in Lee et al. 2021, which is a real fun figure because we obtained a couple of VLPs from various serotypes from Purbright Institute, who are really good at producing them. And the answer is really complex. So that's why I'm saying look at figure 6 in that paper. And actually, it's not that simple. I like the question because I would have hoped that the simple answer would be if it binds 146S, it also binds VLPs. That would imply also that VLPs have the same structure as 146S and would induce good antibodies because they are exactly the same in 3D structure. But I think that's not the case. And there are really interesting things and also things that others can later discover in this field, why this difference in antigenicity that we observe in this figure 6 in that paper, why that occurs. And I've only hinted to it in my presentation for A24 75S, where I also see some differences. But you can see a lot more in that's beyond my understanding.
Okay, that sounds super interesting. I'm going to go and look at figure 6 in that paper because I'm now intrigued. Okay, next question from Amin Asfor. Hi, Amin. Why M170 binding is restricted to only two serotypes, two serotype O? Can we widen the binding by design?
Yeah, fun. We tried it, Amin. Thanks. We did Series 3 randomization. Nishi did. It didn't work. And we also tried tricks like, in essence, I didn't go into structures of VHHs and sequences of VHHs and molecular evolution of recombinant antibodies, which is a big topic in human therapy. And nowadays, a lot can be done. I also did some nice things on this topic by shuffling, DNA shuffling of VHHs that are related to improve particular things like proteolytic stability in the past. But for M170, we chose, we tried this approach for M170 by CDR3 randomization. It didn't work. We got a clone that was not any better. And we tried it for serotype A binding VHHs by DNA shuffling, and also there it didn't work. And my gut feeling is that it's better to go back to phage display selection of original libraries and do a good selection on cross strain binding. That would be my favorite approach if I would do it again. We're going to publish this in our novel paper, my last paper on the topic.
Okay, sounds good. Okay, is the degradation of 146S, does that occur in all types of oils?
I would say yes. We didn't test all, but we did test ESA 206 and a double oil emulsion that was typically of the Lelystad factory before it was sold. And in both it does, and I would be surprised if it would not be a universal phenomenon. Yeah.
Okay. This question is described as being somewhat out of this discussion regarding unpurified FMD vaccines, whether these vaccines would be less effective compared to a purified one. So is a purified
Yeah. I think purification is what you're doing with purification is you're removing proteins. And we have shown clearly that BSA, I've only shown one slide, but in the paper you can see a lot more. And also others publish it often that addition of BSA is good for stabilizing 146S particles. And I think by removing, I think it can be any protein. It's just adding protein. BSA means adding a specific protein. If you purify from BHK cell 1 proteins, then you're removing proteins. So I'm not sure if purification it can prevent proteolysis. I'm not sure about that. I'm not deep into such production processes, but I would certainly not say that purification is always detrimental. It also has is always beneficial. It also has detrimental effects of removing protein that can stabilize.
Yeah. Okay. Another one from Jose. Can you say that since M691 was able to recognize 146S and not 75S, that 75S is conformationally changed significantly?
To keep it simple, yes. That would be my conclusion. That would be fun if you would really could show that also by cryo-EM. That would be fun if you could see the epitope of M170 differences. I'm looking forward to see VLP cryo-EM structures appearing.
Okay, we've got two more questions remaining, and we've got 3 minutes, I think, left in the slot. So, last two questions. Thanks for your nice talk. Can we share some VHHs, or if possible, how can I get some VHHs?
Send an email to me or Dr. Vimian van der Waal. We do sell them, I think, for reasonable prices, like covering the production costs. And if you would be interested in also obtaining VHH sequences, then contact business development of our institute. That's also possible. Yeah.
Okay, thank you. Last question. Is oil emulsion induced degradation of 146S the same for all serotypes?
No, it's not. Yeah, the stability, let's call it the stability after oil emulsification. And this is also in this vaccine paper we published 8 years ago about BSA sucrose and thiomazole, those two. And I think also others more recently have shown studies using sometimes different techniques to analyze stability after oil emulsification. Have shown that there's really differences between strains and differences between serotypes in stability. Each strain has its own intrinsic stability pattern. Yeah, that's a big difference. You have to analyze it for each strain again.
Okay. Okay, that's the end of the formal questions. So I just want to say thank you again, Miguel, for a fantastic talk. I was kind of surprised to hear that you're retiring. Are we going to be able to tempt you back to meetings out of retirement?
Yes, I'm available for that. Certainly. I'm going to do cherry picking then if I like it.
Okay.
But if there's anything fun to discuss and if you want to
So
use
if we put on a GFRA meeting where the agenda looks interesting, we can still tempt you to come.
if somebody pays my costs, because I don't have a salary anymore, that's
Sure.
the thing. But yeah, certainly, I would
Okay,
be certainly eager to do that. Yeah.
brilliant. Okay, thank you very much.
Okay, thank you for the opportunity. It was fun.
Thank you so much for such an engaging presentation, as well as discussion. And thank you, Toby, for moderating this webinar. Thanks also to everyone who joined. Just a quick note, as you log off, you'll see a short survey come up. If you could take just a minute or two to respond to that survey, that we really value and appreciate your feedback in continuing to support and improve this webinar series. And other than that, just stay tuned for more information on our next webinar, which will be happening in March. All right, thank you.
Okay. Bye-bye.